Hello, I'm Adriano, and this is my DNA replication take-home assessment. So I'm going to start with a little overview on DNA. This here is a diagram of DNA. As you can see, it is a double helix, meaning it has 1 and 2 helices. And it is made up of these things called nucleotides. And I've drawn one nucleotide here. Each nucleotide has this 5-carbon sugar, deoxyribose. It has a phosphate group, and it has a base. That base can be either A, T, C, or G. So, what is the structure of DNA? So we each have these nucleotides, and the different bases bind together. A purine binds to a pyrimidine, and there's a specific order to it. A binds to T, and vice versa, and C binds to G, and vice versa. So here you can see these little dotted lines right here. These are all hydrogen bonds between the bases. So the actual DNA backbone itself, that is what makes up these little rungs on which is why DNA is kind of considered a ladder. As you can see, the bases are the rungs on the ladder, and this black part right here, this part is the backbone, which is made up by the sugar and the phosphate, hence its name, the sugar-phosphate backbone. The bonds between the sugar and the phosphate are called phosphodiester bonds, and these are covalent bonds. And it happens between the 3' carbon of the ribose sugar and the phosphate of and the phosphate group of the other nucleotide.
DNA can be used as the hereditary model only because of these specific characteristics. Since DNA is double helixed, not single helix like RNA, it is much more stable since each nucleotide has a cap on it at the end of it, i.e. the other nucleotide. And another important part that makes DNA suitable to be the hereditary information that we pass on is the fact that DNA codes for proteins. So each base, in each set of 3 bases codes for a specific amino acid, and as we all know, a series of amino acids is a protein. So by having these long chains of DNA, we are able to pass on the information that we need to make each protein, which makes it qualifies it to be used as hereditary material.
So DNA replication happens during the S phase of the cell cycle. And this happens because every cell in the body needs DNA. So if the DNA were never replicated, we would not have enough DNA for the trillions of cells that we have. So DNA replication is considered semiconservative. I drew a little depiction of it here. We have 2 strands of DNA. Those are the original 2 strands. After replication, there will be 1 parent and 1 new one. And here we'll also have 1 new one and 1 parent. These would be in the 2 daughter cells of this parent cell. Then, after another round of replication, then this one original parent would go to here, and this one would go to here, and we'd get 2 new DNA strands. And the same thing would happen over here. So this is beneficial because it's energetically cheap, and since it preserves this original strand of DNA, it is less mutation prone because there's always a template to work off of. And even in here, where it doesn't have this original strand, it still contains this strand right here, which, since it's based directly off of that original parent, it can never mess up unless there's something wrong with the actual replication process itself. So now I'm going to go into how DNA replication actually happens. So we start here with a replication fork. That is where the DNA splits. Okay, and we have two strands that I'll be explaining. The leading strand, which is this one here, and the lagging strand, which is this one here. And they both replicate differently. So this happens because DNA is anti-parallel. So if I go back to this drawing here, you can see that one end is facing up and one end is facing down. This end is known as the 5' end, and this end is known as the 3' end, because as you can see here, because it's all based on the number of carbons. So this is the 5' carbon and it's facing up, and here the 3' carbon is facing up. So in replication, DNA polymerase, one of the main enzymes behind replication, it only works from the 5' end to the 3' end. So the actual overall directions of replication will happen like this for the leading strand, because it goes from 5 to 3, and from here in the lagging strand. Because you might be thinking, what do you mean it's 5 to 3? Here, isn't it 3 to 5? No, because this is the 3' end of the template strand, the parent. But the new DNA being made, its 5' end is here. So as the DNA splits, I'm going to erase these bases here just for the sake of the visual. So as the DNA splits, the replication will simply continue in this direction, and this will all happen normally. As it splits, it will continue. There's no breaks in it, and that's just how it works for the leading strand. But on this other side, we need to move in this direction. And since we're moving in this direction, since we're starting wherever it attaches to at that moment in time, there's little splits in it, because originally when it was like this, it's added this much, and then as it wanted to add this much. So it creates these fragments known as Okazaki fragments. Whereas here, it started this way, and as this DNA strand opened up, the DNA just kept on continuing. So now that you understand a little bit more about the directionality of it, I'm gonna explain how the leading strand works. We start with these enzymes. We have topoisomerase and helicase. Helicase is what unwinds the DNA, and that's the enzyme in green. And topoisomerase, the enzyme in yellow, it relieves stress in the DNA, because otherwise, as it's being unwound, it will start to coil in on itself, and that's bad for the DNA. So you can also see this brown thing here. Those are SSBPs, single-stranded DNA binding proteins. They bind to the DNA. SSBPs were not here. The bases would form hydrogen bonds just like they normally do. In helicase, as soon as it unwound them, they would just rebind. So once that has happened, an enzyme called primase adds these RNA primers. RNA primers are these little yellow things here, and they are just a few nucleotides, but instead of being of DNA, they are RNA. This is because our cells were originally made to work with RNA and replicate that instead of DNA. So once the primers have been added, DNA polymerase comes in. DNA polymerase will bind here and move in this direction, adding bases, as you can see here. The blue enzyme is DNA polymerase. It added nucleotides from the 5' end to 3' end, just like that. Then we have this green enzyme, and this green enzyme is exonuclease. Exonuclease will remove these yellow RNA primers, so that way another DNA polymerase, this one is DNA polymerase 1, it will replace them with actual DNA. So this big one right here is DNA polymerase 3, and this one is DNA polymerase 1. They're not to scale. But DNA polymerase 3 is what adds the nucleotides, just the main, and that's the main one that works. And DNA polymerase 1 is what replaces the RNA primers after exonuclease has removed them. So after that happens, I have just moved this back out because that's the lagging strand, and I'll explain that later. So after that happens, you can see there's still no bind between that. There's no of the phosphodiester bond between the backbone of the DNA. So that's where an enzyme called ligase comes in, and it will catalyze the binding of these two, of the carbon, the 3' carbon, and the phosphate group in DNA. So now I'm going to move on to the lagging strand. So the lagging strand works in a very similar way, but it's in the opposite direction. So primase still adds the primers, then polymerase 3 will add the nucleotides in this direction from 5' to 3'. Actually, it'd be from 5' to 3'. And then exonuclease will remove the primers, and DNA polymerase 1 will replace them. Then, as helicase and topoisomerase move on and open more, a new primase will add 2 more primers here, then another DNA polymerase 3 enzyme will move in this direction, adding in bases, adding in nucleotides, and then, just as it always happens, exonuclease will remove the primers and DNA polymerase 1 will replace them. And this continues happening, but as you can see, there's lots of breaks in because of these Okazaki fragments. So because of there's all of these shadows in here, ligase, the same enzyme that does it for the leading strand, will come in and force a phosphodiester bond in the backbone right here, joining them all together. So you might be wondering, where does the energy come from in this reaction? As you can see, there's all these bases being added and all of this binds happening, but where does the energy come from? That is from this thing called DNTP, deoxyribonucleoside triphosphate. This, as you can tell, clearly does look a lot like a regular nucleotide. It simply has these 2 phosphate groups added on, and that's because it is, in essence, just a nucleotide with some more phosphates, because that's what it's made for. When these are what supply not only the energy for DNA replication, but also the nucleotides themselves. So after these phosphates break off and the energy is released, this right here will go on to become part of the DNA. So these come in all the different bases. They have the A, T, C, and G ends to them. So yeah. So the last thing I'm going to talk about is telomeres. So telomeres are a very important part of DNA replication because they fix a major flaw. That flaw is that the enzymes in DNA replication are all very big, so sometimes once everything has been replicated properly, the end of it still won't be replicated. There's just not enough space. So, and we need every single bit of DNA, because if this isn't replicated, then we lose that information which codes for proteins, and our proteins won't function normally without every amino acid in them. So if we lose just the if we lose any amount of DNA, then we won't be able to code for the proper protein because it will have not enough amino acids and won't fold correctly. So how telomeres fix this is they simply extend this. They are nucleotides in a specific sequence that don't code for any proteins, but they're just here to extend that, so that way the enzymes such as DNA polymerase will have enough space to finish adding all of the DNA that we need. So I'm just going to do a quick overview of telomere replication. So once replication has happened, as you can see here, everything is good. We've gotten all of the DNA that we needed, and here's just some replicated telomeres. That's not enough. It's always important to have more telomeres. So here's what we do. So telomerase will extend the telomere sequence, adding more and more telomeres to it, and then we have more space, and DNA polymerase and all of the normal enzymes that deal with DNA replication will go in and replicate these telomeres. So we have, the only difference is the telomerase, which is what binds to here. It is an enzyme, and it will bind here, and it will move in this direction, extending this telomere sequence, so that after it's been extended, we can then extend this the normal way by adding primers, and then DNA polymerase 3 will move in to add the DNA, then removing the primers, then DNA polymerase 1 will fix that, then ligase will join everything together, and just the entire normal sequence to extend this, and then bam, we have all of our DNA, all of the replicated DNA, all of the telomeres, and all of the replicated telomeres. Thank you for watching.